11 months. That is how long a lab I know spent building a knockout cell line using CRISPR-Cas9, designed the guide RNA on an online tool, delivered it into HEK293 cells, ran a T7E1 assay, saw cleavage bands at the right size, confirmed the edit, built an entire paper on those cells, rescue experiments, phenotype data, functional readouts, submitted for publication. One reviewer asked a single question: did you sequence the predicted off-target sites? They sequenced. Cas9 had also cut a tumor suppressor gene on chromosome 7. The cell line they'd built their entire paper on had been silently accumulating genomic damage for 11 months. The paper was retracted.
The enzyme performed correctly.
The enzyme performed perfectly. Every cut it made was exactly where it was directed to cut.
So the guide RNA was designed wrong?
The guide RNA wasn't wrong exactly. It matched the intended target. The problem was that it also matched somewhere else. Just well enough.
It is mildly striking that the most programmable gene editing tool in history fails by being too obedient.
That's exactly it. CRISPR-Cas9 does what you tell it to do. The precision is not in the enzyme, it's in the guide RNA design, and in the validation you run before you trust your result. That's what today is about. Not just how to run the protocol, but the logic behind every step that keeps your experiment from becoming someone else's retraction example.
CRISPR-Cas9 is adapted from a bacterial adaptive immune system. In nature, bacteria incorporate short sequences from invading viral DNA into their own genome, the CRISPR array. When that virus returns, those sequences are transcribed into guide RNAs that direct Cas9 to the viral DNA, which Cas9 cleaves and destroys. In the lab, we've simplified this to two components: the Cas9 protein, a large nuclease with two independent cutting domains, RuBC and HNH, and a synthetic single-guide RNA, the sgRNA. The sgRNA contains a 20-nucleotide spacer sequence complementary to your target, plus a structural scaffold that anchors it to Cas9. When the sgRNA base pairs with the target DNA strand, Cas9 undergoes a conformational change that activates both nucleus domains simultaneously, producing a blunt-ended double-strand break. Before we continue, one structural requirement that people get wrong. Cas9 requires a specific 3-nucleotide motif in the genome immediately 3' of the target sequence. This is the PAM, proto-spacer adjacent motif. For the most commonly used Cas9 from Streptococcus pyogenes, the PAM is NGG. Cas9 checks for this sequence before interrogating the sgRNA match at all. Without it, no cleavage occurs regardless of how perfectly your guide sequence matches the DNA. This constraint limits which targets you can choose. Your 20-nucleotide spacer must terminate at a genomic NGG. Mechanistically, Cas9 does not scan every nucleotide in the genome sequentially. It interrogates DNA at PAM sites first, sliding along the duplex, pausing at every NGG, locally unwinding a short region, and checking the first few nucleotides of the adjacent sequence for sgRNA complementarity. If they match, Cas9 continues unwinding and fully interrogates the site. If they don't, it moves on. This PAM first search is why off-target cutting rates are lower than you'd expect for a protein that tolerates 5 mismatches. Most of the genome is never fully interrogated at all.
Subscribe. The patent licensing fees for commercial CRISPR applications run into the tens of millions annually. The Broad Institute and UC Berkeley went through 5 years of litigation over who invented this. The least you can do is click a button. It costs nothing and funds more of this.
Equipment. Start with your Cas9. You have two main delivery formats: a plasmid that expresses Cas9 and your sgRNA together inside the cell, or a pre-assembled ribonucleoprotein complex, RNP, where Cas9 protein is loaded with your sgRNA in vitro before delivery. Use RNP. It's faster to act, faster to degrade, lower persistent expression, lower off-target footprint, because the complex is cleared within 48 hours. The only reason to use plasmid-based Cas9 in 2026 is cost.
Pre-assembled RNP from a commercial supplier costs around €300 per experiment. Plasmid approach is 40. Unbiased off-target sequencing to validate a plasmid experiment runs about 2,000. RNP is the cheaper option. I've done the arithmetic.
Your sgRNA. Design a 20 nucleotide spacer targeting your sequence of interest, immediately 5' of an NGG PAM in the genome. Use CRISPR or IDT's tool, something that gives you both an on-target efficiency score and a specificity score showing predicted off-target sites genome-wide. You want specificity above 50. High efficiency with low specificity is exactly how you generate the paper we opened with.
I designed mine on a free website. It just asked for the target sequence and gave me one score. It said high efficiency.
Did it show you off-target predictions?
No, it said the score was very good.
That guide RNA is a liability until you validate it independently. Redesign it on CRISPR before you order synthesis. That takes 10 minutes.
Off-target cleavage occurs when the sgRNA base pairs imperfectly with a non-target genomic sequence. SPCS9 tolerates up to 5 mismatches between the guide and the DNA, depending on their position. Mismatches in the seed region, the 8 to 12 nucleotides immediately upstream of the PAM, are least tolerated and most damaging to efficiency. Mismatches in the distal region, nucleotides 13 to 20 from the PAM, are tolerated much more. An off-target site is defined by sequences with good proximal matching even if the distal region diverges. A tool that scores efficiency without scoring off targets is giving you half the information. The 20 nucleotide spacer length is empirically optimized. Shorter spacers reduce on-target efficiency. There isn't sufficient hybridization energy to stably engage the target. Longer spacers don't improve it. 20 nucleotides provides the thermodynamic stability needed for tight target binding while remaining short enough that a single mismatch has meaningful energetic cost. This is why you design at exactly 20, not 18, not 22.
It is mildly ironic that the part of the guide RNA that matters most for specificity is also the part that matters most for efficiency.
Delivery vehicle. For cells in culture, your main options are lipid nanoparticle transfection, lipofection, or electroporation. Lipofection is gentler on the cells, lower efficiency. Electroporation is harsher, more cell death, significantly higher delivery. For hard-to-transfect cells, primary T cells, stem cells, neurons, electroporation or nucleofection is standard. Do not guess pulse conditions. Look up the validated condition for your exact cell type from the instrument manufacturer and use it precisely.
Lipofection encapsulates the cargo in a lipid nanoparticle that is taken up by the cell via endocytosis. Endosomal escape is the rate-limiting step. If the nanoparticle cannot release its cargo before lysosomal degradation, delivery fails. For RNP specifically, endosomal escape is often incomplete, which is one reason electroporation, which bypasses endosomes entirely by direct membrane permeabilization, gives substantially higher delivery efficiency for protein nucleic acid complexes. If lipofection is giving you low editing, the problem is almost always endosomal trapping, not Cas9 activity.
Donor template is only needed for precise edits. For gene knockouts, you don't need one. NGG, the error-prone repair pathway the cell uses when a double-strand break has no repair template, introduces random insertions and deletions at the cut site, disrupting the reading frame. Gene disrupted. If you want to install a specific sequence, correct a point mutation, insert a reporter gene, you need homology-directed repair and a donor template. Single-stranded oligonucleotide for small edits up to about 50 bases. Double-stranded plasmid for larger insertions. HDR efficiency is significantly lower than NHEJ and only works efficiently in dividing cells.
HDR requires the cell to be in S or G2 phase of the cell cycle when the sister chromatid is available as a homologous repair template. In postmitotic cells, neurons, terminally differentiated myocytes, HDR efficiency is near zero. This is a fundamental constraint, not a technique limitation. It's why base editing and prime editing have become important alternatives. They chemically modify DNA without introducing a double-strand break and therefore don't require HDR. They work in non-dividing cells. If your target cell type isn't dividing, classical Cas9 knockout still works fine, but precise insertion does not.
Protocol. Step 1. Design and validate your sgRNA. I want 3 things before you order synthesis. Specificity score above 50 on CRISPR. No predicted off-target sites with fewer than 3 mismatches anywhere in the genome. And no predicted off-target sites with fewer than 4 mismatches in any coding region. Meet all 3 or redesign. This is not optional for anything you're publishing. Step 2. Prepare your RNP complex. Mix Cas9 protein with your sgRNA at equimolar ratio in RNase-free buffer. Let it incubate at room temperature for 10 minutes. This is the complexing step, giving Cas9 time to load the sgRNA into its binding groove. Keep it on ice after complexing. Deliver it the same day you make it. Do not store overnight.
I made mine yesterday and put it in the fridge.
For how long?
About 14 hours. It was very cold.
That RNP is compromised. RNases don't stop in the fridge. They slow, but they don't stop. 14 hours in a standard buffer in a shared fridge with RNase contamination from your bench, your tubes, your hands. That sgRNA has degraded. You're delivering a Cas9 that's loaded with fragments, not a functional guide. Your editing efficiency will be low or 0, and you won't understand why until you've wasted a transfection.
RNA is intrinsically susceptible to RNase cleavage, and RNases are extraordinarily stable, surface-persistent enzymes present on hands, benchtops, standard plastic labware, and any water that wasn't specifically prepared RNase-free. When the sgRNA component of an RNP degrades, Cas9 retains DNA binding activity but loses targeting specificity. The result is a protein that interacts with DNA nonspecifically, producing background signal with no productive cutting. Always use dedicated RNase-free tubes and reagents, change gloves frequently, and prepare the RNP within an hour of delivery. Freshness is not a preference, it's the protocol.
It is mildly reassuring that a molecular machine capable of rewriting the human genome is failed by not changing gloves.
Step 3. Deliver to cells. For electroporation, load your cell RNP suspension into the cuvette or cartridge specified for your protocol. Run the validated pulse. Plate your cells immediately after. Do not let electroporated cells sit in the cuvette while you do something else. Membrane disruption is temporary, but dead cells accumulate fast, and they take the viable ones with them if you wait too long.
Electroporation applies a brief high-voltage electrical pulse that transiently permeabilizes the plasma membrane, creating pores large enough for macromolecular cargo to enter the cell by diffusion. Pulse parameters, voltage, capacitance, pulse duration, are empirically optimized per cell type and must be used exactly. Overvoltage kills the cells. Undervoltage means insufficient membrane permeabilization and delivery fails. Neither outcome generates an error message. Both look identical on your editing readout, no editing, and are diagnosed only by checking viability alongside efficiency.
Step 4. Incubate and collect. For RNP delivery, peak editing occurs at 24 to 48 hours post-transfection. After that, collect your cells, extract genomic DNA, and validate.
I collected at 6 hours. I needed the cells for another assay.
At 6 hours, the editing may still be in progress, and you're collecting during an active DNA damage response. The NHEJ indels are not fully stabilized. Your T7A1 result at 6 hours will underestimate your actual editing efficiency, and you might conclude the experiment failed when it didn't. Wait 24 hours minimum.
NHEJ-mediated repair occurs in stages. The double-strand break is sensed within minutes, but the error-prone ligation, fill-in, and indel incorporation process continues for many hours. Collecting cells at 6 hours captures an intermediate state where some alleles are repaired, some are still broken, and some are being actively processed. Your T7E1 gel will show weak or ambiguous cleavage bands, and your Sanger sequencing will show a mixed, partially overlapping chromatogram that's difficult to interpret. This is a timing artifact, not a guide design failure. 24 hours is the minimum collection point for RNP experiments.
Step 5. Validate. Run the T7E1 assay first. PCR your target locus from extracted genomic DNA, then denature and reanneal the amplicon slowly. This step is critical, and most protocols underspecify it. The slow reanneal allows edited and unedited PCR products to form heteroduplexes. Digest with T7E1 endonucleus, which cleaves DNA at mismatched positions in those heteroduplexes. Run on an agarose gel. Cleavage bands at the expected sizes confirm editing is occurring.
What good data looks like. The T7E1 gel shows your full-length uncut amplicon band, plus 2 smaller cleavage product bands at sizes that sum to the full amplicon length. Band density ratios give you an editing efficiency estimate, the proportion of cut to total. Above 30% in HEK293 cells with a well-scoring guide and RNP delivery is a solid, first-pass result. Confirm with Sanger sequencing of the locus. At the edit site, you'll see overlapping chromatogram peaks beginning at the expected cut position, the signature of a mixed population of alleles. Tied analysis of that chromatogram quantifies the efficiency and shows the distribution of indel sizes. The indel distribution matters. NHEJ repair is stochastic. Each allele generates a different insertion or deletion. The most common outcomes are single nucleotide insertions and short deletions of 1 to 5 bases, all of which cause frame shifts in coding sequences. However, a minority of repair events produce in-frame deletions, multiples of 3 nucleotides, which can leave a truncated but partially functional protein. If complete protein elimination is essential to your experiment, confirm with a functional readout or Western blot, not sequencing alone.
What failure looks like. T7E1 gel with only the uncut band means no detectable editing or editing below 5%. Work backwards. Was the RNP fresh? Was delivery confirmed? Did you check viability? And did you include a fluorescent cargo control? If delivery was good and editing is still absent, go back to guide design. Redesign using a different target site before you change the protocol. Applications. Gene knockout in cell lines for mechanistic studies. Genome-wide knockout screens using arrayed or pooled guide libraries. Deliver thousands of guides. Read out which genes drive a phenotype. Disease modeling. Install or remove specific mutations in relevant cell lines. Gene correction, and this is not theoretical anymore. Casgevy, approved in 2023 for sickle cell disease and beta thalassemia, uses CRISPR-Cas9 to disrupt the BCL11A erythroid enhancer and reactivate fetal hemoglobin production in patient stem cells. First, CRISPR therapy, approved by the FDA and EMA. It works.
Casgevy is priced at $2.2 million per patient. If that number sounds outrageous, a lifetime of sickle cell management, including transfusions, hospitalizations, and organ damage, runs to a comparable figure over 40 years. The economics are not as irrational as they look. They are still very concerning.
The CRISPR platform has expanded considerably beyond classical Cas9 cutting. Base editors pair a catalytically dead or nicus Cas9 with a DNA deaminase enzyme to chemically convert one nucleotide to another, adenine to guanine, cytosine to thymine, without introducing a double-strand break. Prime editors use a nicus Cas9 fused to a reverse transcriptase and a pegRNA that templates new sequence directly into the nick site. Both approaches broaden the range of editable mutations while avoiding the chromosomal instability associated with double-strand breaks. And separately, Cas12 and Cas13 are being deployed as ultrasensitive diagnostic tools. When Cas12 or Cas13 recognizes its target, it becomes a nonspecific nuclease, cleaving nearby reporter molecules and generating a detectable signal. The Sherlock and DetectTR platforms are built on this. They don't edit, they report. Same foundational biology, completely different application.
It is mildly characteristic of molecular biology that the tool responsible for the most significant therapeutic advance of the decade is now mainly famous for its broken derivative forms.
About the editing project I've been running in my shed.
I am not going to be associated with this.
I want to be clear that I am using an RNP complex. Pre-assembled, fresh each session, properly complexed. I've read the literature. My guide has a specificity score of 71 on CRISPR.
I'm also not going to be associated with this.
I've been systematically editing a specific locus in a plant species that I will not identify for legal reasons that I will also not specify. I have 43 plants currently being phenotyped. We are on batch 8.
Batch 8. T71 came back at 68% efficiency. I planted the F2 generation this morning. The shed is at capacity.
68%, that's clean work.
It is mildly notable that the evidence of responsible methodology was the first thing offered in defense of an unlicensed experiment.
Closing tip. The seed region of your sgRNA, the 8 to 12 nucleotides directly upstream of the PAM, is the zone where guide RNA performance is decided. A mismatch there kills on-target efficiency. But it's also the zone where a single near match can open an off-target site that in silico tools will not flag with confidence. Before you order your guide, take those 8 proximal nucleotides alone and run a blast search against your organism's full genome. Not the prediction tool, not the full 20 nucleotide spacer, just the 8. Look at every hit in the top 20. If anything in a coding sequence matches 7 or 8 of those 8 nucleotides, you have an off-target risk that no algorithm caught. That check takes 5 minutes. The validation it replaces takes months. Method Monday.